Good morning, student. Today we will discuss about the gene therapy. Okay, gene therapy is very important thing, children, because gene therapy is a collection of different methods by which we can correct the defected gene. Okay, if we want to correct the defected gene in child or if in an embryo, that time this comes under the gene therapy. Okay, so the defective gene has to be identified. Afterwards only the particular remedy of it can be transferred inside of the patient. How is it so? Now we will discuss it about this process. Okay, the correction of genetic defects involve delivery of normal gene into the individual or embryo. This line is very important, children. Okay, and compensate for the non-functional gene. Means if you want to correct any gene, suppose this individual is having one defect, this gene defect. Okay, firstly you have identified it leads a genetic disorder in that individual. That time what you will do, you will firstly identify it, and this is the defective gene, so you have to search the normal gene first. Then only with the help of genetic engineering you just inject it inside the bone marrow of this patient. Okay, which gene? Normal gene. Then when this normal gene is injected in the bone marrow, that time only it just combined with the whole genome of the individual and lymphocytes especially, and then only they work normally. This is a one process. And second process, if in early embryonic stage this disorder is identified, okay, that time only the normal gene is introduced into the early embryonic stage, and this is the permanent cure. I am telling you, this is not permanent cure, children, because for such type of therapy, for such type of treatment, it requires, because this process is not immortal. Okay, this process has to be rewind again and again. Okay, so regular transfusion of such type of treatment is required for the life of this patient. But if you do this treatment in an early embryonic stage, that time it can be curable. Okay, one example is given in NCERT book, and this example is very important, children, that's why I am sharing it here. This example is in the first clinical gene therapy was given in 1990. Okay, in 4-year-old girl, and the deficiency is the adenosine deaminase deficiency. Means his body is not able to produce one protein. Okay, and this protein name is adenosine deaminase. Okay, this enzyme, children, this enzyme adenosine deaminase, this enzyme is very crucial for the proper functioning of immune system. So that time what happened, that those children who are suffering from this disease, they have to be keep inside a one room only. Okay, this particular patient can't go outside because immune system is very low. If this patient gets a normal exposure of environment, this patient get infection so easily. Okay, so that's why this child should not go anywhere. This child is always have to be in a one room only under the observation of doctor, so life was very difficult. Okay, so for this the scientist has searched one alternative of the treatment, and the alternative of this treatment is of they did ADA deficiency can be cured by bone marrow transplantation. Okay, so firstly it can be cured by bone marrow transplantation, or other way, the gene who is responsible for this deficiency, this gene has to be replaced, or functional ADA is given to the patient. Functional ADA means, children, normal ADA. In people, normal ADA gene, this which I told you already, the normal gene has to be transfused in the patient body. Okay, so as a first step towards gene therapy, the lymphocyte, children, please try to understand it. This is a very important thing. The lymphocyte from the blood of the patient are grown in a culture outside the body. Firstly, what we have to do in a first step, the lymphocyte of patient body has to be isolated and cultured it inside the cultured outside the body of patient. Okay, now lymphocytes are culturing here. These are the lymphocyte and they are culturing here. Okay, then after the culture formation, okay, after the culture formation, what happen, a functional ADA or cDNA, children, last day also I have told you, direct mRNA we can't develop inside the prokaryotic cell. So firstly we have to make its complementary DNA. Okay, because inside the vector also, vector can't produce different enzymes like eukaryotic cell. Okay, so that's why we have to directly just transfer cDNA. Okay, because we are using here retrovirus vector. Children, different vectors are here like bacterial vector or yeast vector, and one vector is of retroviral vector. Okay, so we use this cDNA, ADA cDNA, adenosine deaminase cDNA. Okay, the functional adenosine deaminase cDNA has to be transferred inside these lymphocyte. Okay, and these lymphocytes carry what, functional ADA. So that time directly we are just returning it inside the patient. Now again that patient only, now this patient is having lymphocyte along with the functional ADA cDNA. Okay, now this patient is having functional ADA cDNA. So that time it provide the what, power, or it provide the capacity of this patient to produce these what, this protein. Okay, which protein it is, adenosine deaminase, by which this person gets the immunity booster protein. Are you getting my point? But as I told you, these cells, these lymphocyte cell, which lymphocyte cell, which is transfused now, okay, which is just इंजेक्टेड नाउ फ्रॉम द आउटसाइड, दे आर नॉट इमॉर्टल. सो दीज सेल्स जो लिम्फोसाइट बाहर बनाई थी ना अभी जेनेटिक इंजीनियर से, जिसमें एडीए फंक्शनल है, इट हैज़ टू बी ट्रांसफ्यूज अगेन एंड अगेन इन अ रेगुलर इंटरवल ऑफ टाइम, बिकॉज दे आर नॉट इमॉर्टल. वो जब तक एक्टिव है तब तक बॉडी सही काम करेगी, जैसे उनका नंबर कम हुआ बॉडी फिर से ख़राब हो गई. ओके, सो दे आर नॉट इमॉर्टल, सो देयर ट्रांसफ्यूजन is very necessary in a regular interval of time. Okay, I hope this is clear. But as I told you, it can be curable. There's a permanent solution is there if we identify disorder in a embryonic stage, early embryonic stage, and treatment has to be given at that time. So children, if gynecologist identified any kind of chronic disorder while ultrasound of an unborn baby, so that time gynecologist only suggest for the Doppler type of ultrasound. Okay, by the advanced ultrasound we can easily identify that this embryo is having any kind of disorder. Okay, and the treatment are there in a early stage of embryonic development. Okay, I hope this is clear. Gene therapy is clear. Now there is a several uses of genetic engineering. Now we will discuss about the several practical application of genetic engineering, like diagnosis of disease or molecular diagnosis. Okay, genetic as well as infectious disease can be diagnosed by using DNA technology, and we know it with the help of PCR, with the help of ELISA. Okay, ELISA, children, please learn this name. ELISA means enzyme linked immunosorbent assay, and this technique is especially used for AIDS, for the identification of HIV virus. Okay, enzyme linked immunosorbent assay, enzyme linked immunosorbent assay, I am telling you again and again this name because it is very important. Many times it has asked in board examination also, in competitive examination also. Okay, so PCR and ELISA, these are the recombinant DNA technology by which we can identify any kind of disease very easily. Okay, by the serum, by the urine, okay, by the blood, as I told you. Okay, by these mediums we can easily identify different disease, and it is possible because of the genetic engineering. Okay, presence of different pathogen like bacterial infection, viral infection, it can also be diagnosed by the different kind of genetic engineering tools like PCR. PCR is very important, children. By the amplification of particular DNA, we can easily identify that it is defected DNA or it is this is the normal DNA. Okay, so detection of HIV, as I told you, it only done by the ELISA. I think it is clear. Or single stranded DNA or RNA is a radioactive molecule that hybridize the complementary DNA. Okay, with the help of autoradiography also we can identify the normal or infected DNA. Okay, so children, the main functioning of ELISA, the principle of ELISA is antigen antibody interaction. Okay, infection by pathogen can be detected by the presence of antigens and or by detecting the antibodies synthesized against the pathogen. Clear? Means antigen जो बाहर से आया, एंटीबॉडी जो बॉडी ने बनाई. ओके, इफ दे बोथ आर प्रेजेंट इन द बॉडी, सो इट मीन्स दैट इट प्रूव्स दैट इन्फ्लेमेशन इज देयर. Okay, and due to this inflammation disease is caused. So we can identify different disease if we know which antigen is secreting and which antibodies are developed by our body. Okay, so by this method only we can identify the different kind of, and we can diagnose the different diseases. So gene therapy or genetic engineering we can use for the production of genetically modified organism, GMOs. Okay, and different kind of crop protection method or depollution of environment, or many things are there where we use genetic engineering. Okay, I hope today's topic is clear. Now in next topic we will cover up the last topics of biotechnology. These are the biopiracy and all, and afterward we will start the next unit, it is of ecology. Thank you, children.